All right, let's get our gloves on and beakers out because today we're diving deep into the chemistry of 5-MeO-DMT.
Yeah, getting into the nitty-gritty of actually synthesizing pharmaceutical-grade 5-MeO-DMT.
We all know about the growing interest in its potential therapeutic uses.
But today, we're putting on our chemist hats and focusing on the molecule itself.
It's going to be a fascinating journey for sure.
Oh, absolutely.
So, first things first, why even bother synthesizing it?
Why not just, extract it from the Sonoran Desert toad?
Exactly. It's famous for having loads of this stuff naturally, right?
Right, but there are a few reasons why relying on toad extraction just wouldn't cut it for clinical use.
Okay, I'm guessing consistency and purity are probably at the top of the list.
You got it. Imagine trying to run a clinical trial where the active compound is all over the place from batch to batch.
It'd be chaos.
You wouldn't be able to tell if the effects you're seeing are from the compound or just variations in the natural extract.
Like you couldn't compare results reliably or anything.
Exactly. And then you've got the ethical considerations.
Oh yeah, harvesting from toads raises all sorts of questions about animal welfare.
Not to mention the sheer difficulty of controlling for all the other compounds present in those secretions.
So synthesizing it gives you that control you need for clinical trials.
Exactly. And the researchers specifically aimed for a stable, injectable form suitable for those trials, which meant going beyond just making the 5-MeO-DMT itself.
Which is how we end up with 5-MeO-DMT Succinate.
That's the star of the show.
So why a salt form? And why succinate specifically?
Well, remember, for clinical use, you need a compound that ticks all the boxes. Stable, water-soluble, safe.
Right, especially for injections.
Now, 5-MeO-DMT freebase, the form you find in nature, doesn't quite cut it on those fronts.
It's not the most well-behaved molecule then.
Not really, not very water-soluble, and it's prone to degrading over time, so forming a salt helps address those issues.
So it's like making it more pharmaceutically friendly.
Precisely. Reacting it with an acid, in this case succinic acid, creates a much more stable and soluble salt form.
Okay, that makes sense. But why succinate over, say, hydrochloride? Isn't that more common for pharmaceutical salts?
It is. And they actually looked at other options like hydrochloride and fumarate, but succinate won out for a few key reasons.
Okay, I'm all ears. What made succinate so special?
Well, for one, it's incredibly stable, even under conditions like sterilization, which is a must-have for any pharmaceutical product.
Right, you don't want your drug falling apart before it even gets to the patient.
Definitely not. And then there's this interesting quirk with fumarate. It's what we call a Michael acceptor, which means it can react with amines under certain conditions.
Oh, and 5-MeO-DMT, being a tryptamine, has an amine group.
Exactly. So there's potential there for unwanted reactions, especially with heat.
So it's a risk you don't want to take with a drug that needs to be precise and predictable.
Exactly. Succinate, on the other hand, is much less prone to those kinds of reactions, so it's a safer bet overall.
Gotcha. So they really thought this through, huh?
Oh yeah, they even confirmed the succinate salt stability using all sorts of fancy techniques, like X-ray powder diffraction.
So we can be confident that it's the real deal.
For sure. Now, let's shift gears a bit. How did they actually make the 5-MeO-DMT freebase in the first place?
Yeah, where do you even start with something like that?
They explored 3 different synthetic routes.
Always good to have options.
Definitely.
Sure.
First up was a modified Eschweiler-Clark reaction.
Sounds pretty straightforward.
Yeah, it seemed simple enough on paper, but they ran into a big snag with byproducts.
What kind of byproducts are we talking about?
Stuff that was really hard to get rid of during purification.
I can imagine that's a nightmare.
It would have been a huge headache to scale up, so they moved on to route number 2.
Okay, what was behind door number 2?
The good old Speeder-Anthony tryptamine synthesis. It's a classic.
What was the problem with that one?
The final reduction step used lithium aluminum hydride. That stuff can be a bit, shall we say, temperamental.
Temperamental in what way?
Well, for one, it can burst into flames if you look at it wrong, especially in large amounts.
Not exactly ideal for mass production.
Definitely not. Plus, the reaction needed really specific conditions to work well, so it wasn't the most practical route.
Okay, so 2 routes down. What was their final strategy?
This is where things get really interesting. They went with the Fischer indole reaction.
Another classic, right?
Yep. It's a one-step synthesis and can be done under pretty mild conditions.
So less room for error, safer, and potentially easier to scale up.
All good things. But even this route had some early hiccups.
What happened?
Their initial attempts gave them lower yields than they wanted, plus they had some impurities to deal with.
Ah, so even the easy route wasn't a walk in the park.
Nope. But here's the cool part. They found that using acetonitrile as a co-solvent made a huge difference.
Acetonitrile. Why acetonitrile?
That's a great question. It wasn't just a random choice. They systematically tested different co-solvents and found that acetonitrile was the sweet spot.
So it boosted the yield and helped with purity.
Exactly. And they didn't stop there. They went on to tweak all sorts of other reaction parameters, fine-tuning everything.
That's dedication.
It's a real testament to their attention to detail. And, it's interesting that they chose to go the synthetic route, even though 5-MeO-DMT is readily available in nature.
Yeah. Why was that?
Well, extracting it from toads has a lot of downsides. Ethical concerns, for one.
Right. We talked about that earlier.
But also, the concentration and purity can vary wildly depending on the toad, its environment, all sorts of factors.
So for clinical use, you need something more reliable.
Exactly. The synthetic route gave them the control to create a consistent, high-purity product.
Which is exactly what you need when you're dealing with potential pharmaceuticals.
Absolutely. So they nailed down their optimized synthesis, but purifying it to pharmaceutical standards is a whole other challenge.
That's where the real magic happens, I guess.
You could say that.
Yeah.
And that's what we'll be diving into next.
Can't wait to hear about that. I'm sure there are tons of tricks and techniques involved.
Yeah. Oh, yeah. They get pretty creative with it.
All right, well, let's take a quick breather, and then we'll jump right into the purification process.
Back again, ready to get down and dirty with purification.
Ah, yes, the unsung hero of organic chemistry.
It really is. You can have the most elegant synthesis in the world, but if you can't purify your product...
It's all for nothing.
Exactly. Especially when you're aiming for pharmaceutical grade.
The standards are sky high.
No room for error. So, let's get into it. The first hurdle they faced was a sneaky little byproduct that popped up during the workup.
During the what now?
Oh, right, sorry, the workup. It's basically everything you do after the reaction is finished to isolate and purify your product.
Like separating the gold from the dirt.
Exactly. And in this case, the dirt was a rather unexpected guest.
Okay, spill the tea. What was causing trouble?
They started out using dichloromethane to extract the 5-MeO-DMT.
Dichloromethane. Pretty standard solvent, right?
Oh yeah, it's a workhorse in the lab, but it turns out it can be a bit reactive with certain compounds.
You mean like amines?
You got it. And remember, 5-MeO-DMT has that tertiary amine group.
Right, from the tristamine backbone.
Exactly. So dichloromethane was reacting with that amine to form quaternary ammonium salts.
And those were showing up as impurities.
Yep. And they were a pain to separate from the 5-MeO-DMT. So dichloromethane was out.
Back to the drawing board.
They had to find a new solvent that wouldn't cause those side reactions.
So what did they end up using?
After some testing, they landed on 2-methyltetrahydrofuran, 2-methec-F for short.
Get your name.
It's a mouthful, right? But it worked like a charm.
What made it so much better?
Well, first off, it's much less reactive with 5-MeO-DMT, so no more quaternary ammonium salts.
Problem solved.
Plus, it's considered a greener solvent, so bonus points for that.
Always a good thing.
Right. But switching solvents was just the first step. They still had to deal with some pesky dimer impurities.
Dimer impurities? What are those?
It's when two 5-MeO-DMT molecules decide to stick together.
Like molecular buddies?
Pretty much. And these dimers were really hard to get rid of using traditional methods.
Why was that?
They have very similar properties to the 5-MeO-DMT itself. They tend to crystallize together.
So, like trying to separate two types of sand that look and feel the same.
Exactly. It's a tough one. But they had a trick up their sleeve.
Oh. I love a good chemistry trick.
They used silica gel filtration.
Silica gel. Like what's in those little packets that come with electronics?
That's the one. But in this case, they're using it to exploit the subtle differences in polarity between 5-MeO-DMT and the dimers.
Polarity. Can you break that down for me?
Sure. Polarity is all about how electrons are distributed in a molecule.
Some molecules are more electron hungry than others, basically.
You got it. And silica gel happens to be a polar material, so it attracts polar molecules more strongly.
Okay, and 5-MeO-DMT is?
Less polar than those pesky dimers. So when you run the mixture through a column of silica gel,
the dimers stick to it more strongly, and the 5-MeO-DMT flows through.
You got it. It's a clever way to separate them based on that tiny difference in polarity.
So silica gel saves the day again.
Pretty much. And with the dimers out of the picture, they were ready for the final step. Salt formation.
The moment of truth.
Time to turn that purified freebase into the stable succinate salt we've been talking about.
I bet there's more to it than just mixing them together.
Oh yeah, they weren't about to cut any corners. They wanted a perfectly pure and crystalline final product.
So how'd they do it?
First, they dissolved the purified freebase in methanol.
Methanol, another classic.
It's a good one. Then they added just the right amount of succinic acid.
Like a delicate dance of molecules.
That's a great way to put it. They let it react under very controlled conditions, precise temperature, precise time.
I can imagine even a small change could mess things up.
Absolutely. They were aiming for a very specific crystal structure.
So they've got their reaction mixture. What happens next?
Well, they ended up with a solution of 5-MeO-DMT Succinate, but it wasn't quite ready for prime time.
More purification steps.
You know it. But first, they ran into an interesting little challenge. The color of the product was slightly different from batch to batch.
Even though the starting materials were supposed to be pure.
Yep. Turns out even trace amounts of impurities can affect the color.
So what did they do about it?
They brought out the big guns. Activated charcoal.
Like the stuff you use to filter water.
The very same. It's incredibly porous, like a molecular sponge, so it can grab onto those tiny impurities that cause color variations.
So they just added it to the solution and filtered it out.
Pretty much. Simple but effective.
I love it when a plan comes together.
Me too. Okay, so now they have their decolorized solution of 5-MeO-DMT Succinate. Time for the grand finale?
Let's hear it.
They concentrated the solution, basically removing most of the methanol, which left them with a thick slurry of the salt.
Slurry. That's a fun word.
It's a bit technical, but basically it's a mixture of solid particles suspended in a liquid.
Okay, I get it. So what was the point of making a slurry?
This is where they use the power of crystallization. They slowly cooled the slurry, and as it cooled, the 5-MeO-DMT succinate started to form beautiful crystals.
And the impurities.
They stayed dissolved in the remaining liquid, so they were easily separated.
Crystallization really is a chemist's best friend.
It's a powerful tool. So they filtered out those shiny crystals, gave them a quick wash with acetone to remove any last traces of anything unwanted, and dried them.
And there you have it. Pharmaceutical grade 5-MeO-DMT succinate.
Ready for its close-up.
What an incredible process. So much precision and attention to detail.
It's amazing what it takes to create something truly pure and reliable.
Well, we've covered a lot of ground, but there's still one more piece of the puzzle.
Ah, yes, the N-oxide degradant.
Right, that little metabolite we mentioned earlier.
Even though it's naturally occurring, the researchers didn't just brush it aside.
They wanted to know everything about it.
And that's where we'll pick up next time.
Can't wait to delve into the world of N-oxide characterization. It's always fascinating to see how these small details can have such a big impact.
Absolutely. It's all part of the journey.
And we're back, ready to tackle the final chapter in our 5-MeO-DMT saga.
The N-oxide degradant, taking center stage.
Even though it's a natural metabolite, the researchers really went all out in characterizing it.
They didn't want to leave any stone unturned.
So why all the fuss about something that our bodies already produce?
Well, for one, when you're talking about pharmaceuticals, you need to know exactly what's in your final product, even the minor players.
FDA is pretty strict about that, right?
Yeah, yeah. And beyond the regulatory side, understanding the N-oxide gives us valuable clues about the drug's stability and how it might degrade over time.
So it's about more than just ticking boxes for the FDA. It's about understanding the molecule inside and out.
Exactly. So how did they go about getting to know this N-oxide degradant?
I remember reading that they actually synthesized it separately.
They did. It might seem counterintuitive to create the impurity on purpose, but it's a common strategy in pharmaceutical chemistry.
What's the logic there?
To really get a handle on an impurity, you need a pure sample of it. That way you can analyze it without interference from other compounds.
So it's like having a clean fingerprint to compare against.
Exactly. Once they had their pure N-oxide, they could unleash the full power of analytical chemistry to figure out its structure.
I'm guessing this is where the heavy hitters like NMR and mass spec come in.
You got it. They use a combination of high-resolution mass spectrometry and NMR spectroscopy.
Powerful tools for sure. What kind of information did they get from those?
Well, high-resolution mass spec gave them the exact mass of the molecule, confirming what atoms it was made of.
So they knew exactly what they were dealing with.
Yep. And then NMR gave them a 3D map of how those atoms were arranged within the molecule.
So they had rock-solid proof that they had isolated and characterized the N-oxide.
Absolutely. And that thorough characterization served several purposes. For one, they could now accurately measure how much N-oxide was present in their final product.
Important for making sure it met purity standards.
Exactly. But it also gave them a deeper understanding of how 5-MeO-DMT breaks down and what factors might influence that degradation.
So it's about predicting and controlling what happens to the drug over time.
Precisely. Knowledge is power, especially when you're developing a new pharmaceutical.
This whole deep dive has been incredible. We've seen every step of the journey, from choosing the right synthesis route to purifying the final product to pharmaceutical standards.
It's amazing how much work goes into creating a drug that's safe, effective, and reliable.
And it all starts with a deep understanding of the chemistry involved.
Absolutely. From the main molecule to the smallest impurity, every detail matters.
Well said. Well, folks, that brings us to the end of our deep dive into 5-MeO-DMT chemistry.
It's been a wild ride through the world of organic synthesis, purification, and characterization.
We've seen firsthand the challenges and triumphs of creating a pharmaceutical-grade version of this fascinating compound.
And hopefully we've sparked some curiosity about the incredible power of chemistry to unlock the secrets of these molecules.
Thanks for joining us on this journey. And until next time, keep exploring the world of chemistry.
Keep asking questions and never stop learning.